You can start, ma'am.
Okay.
Ma'am, we are live now.
Okay. So, hello students, just give me a few seconds. Good evening, everyone. I'm going to share my screen. So, hello, everyone. I hope you can see my screen. Neha, can you give me the YouTube link so that I can have a look at students' chat?
Let me send you a link, ma'am.
Okay. So, as your INI CET exam is approaching, so it will be highly recommendable for you to do the PYQs. And I am here with you on eGurukul to revise these questions and these topics actually. So let's start and let's do whatever we can do in this less time. And I hope I will be able to stimulate you to study these topics. And immediately after the session, you will be sitting and revising these topics. So give at least 1 hour after the session to revise these topics. And I will share the annotated PDF of this PPT slide, which I have made. So so this also you can revise. Okay. So first we'll be doing November 22 exam questions. Yeah, the first question is, a baby is being evaluated for delayed developmental milestone. On examination, child has hepatosplenomegaly. So more of clinical questions are asked these days. So be very careful, integrate all the subjects. While studying, integrate all the subjects. Body is not 19 subjects. Okay. So please, whenever you are studying, just keep floating between, flying between one subject to another, right? Okay, let's continue. Microscopy of bone marrow peripheral smear evaluation is shown below. So you can see the image. Can you see this is cotton ball like or crumpled tissue paper appearance, right? When I say crumpled tissue paper appearance, then the diagnosis is very clear to you. So what is the diagnosis? Diagnosis is Gaucher's disease. What is Gaucher's disease? It is most common lysosomal storage disease. In Gaucher's disease, it is a sphingolipidosis. It is a sphingolipidosis. The enzyme deficient is beta glucocerebrosidase. Patient has easy bruisability. So commonly bruises are there. And skeletal abnormalities. Skeletal abnormalities. So very frequently question is asked on sphingolipidosis, especially Gaucher's disease. Do all the lysosomal storage diseases once, okay? So This is Gaucher tissue paper appearance, cell crumpled tissue paper appearance, and the substrate which gets accumulated, because this enzyme is not working. So which substrate will accumulate? Glucocerebroside. Glucocerebroside will accumulate, it will increase. There is no mental retardation in these patients because glucocerebroside is not found in brain, right? So this was Gaucher's disease. Which enzyme deficient in Niemann-Pick disease? Sphingomyelinase. Sphingomyelinase. In Hurler and Hunter, these are mucopolysaccharidosis. Type 1 is Hurler, type 2 is Hunter. From Hunter, we easily remember that this is a male patient, X-linked recessive, and there is no corneal clouding. There is no corneal clouding. Otherwise, corneal clouding occurs in all the mucopolysaccharidosis, right? So we have done the other 3 options also. So do read all these 4 topics: mucopolysaccharidosis, Hurler and Hunter, Niemann-Pick disease, and Gaucher's disease, right? Okay. Next, a baby is brought with complaints of lethargy, poor feeding. During examination, it is noted that urine had a smell of burnt sugar. When burnt sugar is written, your diagnosis should be made quickly. So burnt sugar, maple syrup urine disorder, right? So urine smell is like burnt sugar. This disease occurs due to defect in. This is a defect in the catabolism of branch chain amino acids. So it is branch chain keto acid dehydrogenase, right? Branch chain amino acids in their catabolism, first by transamination reaction, transaminase enzyme, it gets converted to branch chain keto acid, and then branch chain keto acid will form some product. You need not remember the product, but this reaction is oxidative decarboxylation step. This oxidative decarboxylation step is affected in MSUD. This enzyme requires vitamin B1. Any step of oxidative decarboxylation requires vitamin B1, right? So this is affected in MSUD, right? These patients have ketosis. So because branch chain keto acids will be increased, branch chain amino acids will also be increased. So patient has ketosis. There will be increased amount of branch chain amino acids and keto acids in blood and urine of these patients, right? Okay. Next, choose the right sequence of steps in the conversion of cholesterol to testosterone. So let's see on next slide, the conversion of cholesterol to testosterone. Cholesterol gets converted to aldosterone, cortisone also. Here we have to see the enzymes required till the formation of testosterone. So cholesterol to pregnenolone enzyme is desmolase. There's no need to do this full complex pathway of cholesterol to testosterone synthesis. You can just cram this this question a little bit. See, some questions are asked like once in 5 years. So for that, we don't recommend you to waste your time in memorizing such a thing, which is very difficult and very, very high on memory, right? So just cram this question, I will suggest. I'm a very easygoing person. I always like simplified things, and in life also we should do just simplified things, right? So this is desmolase enzyme. Then pregnenolone is formed. Pregnenolone will be converted to progesterone by dehydrogenase enzyme, right? Beta hydroxysteroid dehydrogenase. Then we have hydroxylase enzyme. So then progesterone, 17-hydroxyprogesterone is formed. Then we have lyase enzyme, androstenedione is formed, and then it will be converted to testosterone, right? Important from biochemistry point of view is the dehydrogenase enzyme. Any dehydrogenase will use either NAD or FAD or NADP, right? So NAD will be converted to NADH, FAD to FADH2, NADP to NADPH. This NADH, FADH2 usually required in catabolic reactions and it gives energy. NADPH is used in anabolic pathways. Many times questions are asked on NADPH, right? So This you can remember, cholesterol is a 27 carbon steroid ring, which gets converted to pregnenolone, which is 21 carbons. Progesterone is also 21 carbons. So first 6 carbons are removed from 27 carbon compound gets converted to 21 carbons, right? Then it gets converted to androgens and estrogens. Androgens are 19 carbons and estrogens are having 18 carbons, right? On one side, progesterone gets converted to cortisone, mineralocorticoids, and glucocorticoids also, right? Then all are true about HMP shunt except. So HMP shunt, it gives ribose 5-phosphate and NADPH. NADPH can come from other sources also, but ribose 5-phosphate's only source is HMP. Ribose 5-phosphate can be only obtained from HMP. This is very important, right? Now, so whenever except word is written in the question, be very careful with that question, okay? So all are true about HMP except. True is, it is a source of ribose 5-phosphate, correct? It is a source of acetyl-CoA. See, in HMP, we take 3 molecules of glucose 6-phosphate, 6 NADP. We get 2 molecules of glucose 6-phosphate, 2 molecules of glucose 6-phosphate, 1 molecule of glyceraldehyde 3-phosphate, 1 molecule of glyceraldehyde 3-phosphate, 3 carbon dioxide, and 6 NADPH. So when we get glucose 6-phosphate, it will enter into glycolysis, will go into link reaction, and via link reaction, it will produce acetyl-CoA. So indirectly, it can form acetyl-CoA. Okay, so this can also be considered correct. Option B. It is a source of NADPH. Yes, I told you it is a source of NADPH. It is the major source of NADPH, right? Then it utilizes ribose. It utilizes ribose. This is wrong. Okay, it produces ribose 5-phosphate, does not utilize, right? So answer is D. Okay. Then next, Refsum's disease. Refsum's disease is due to deficiency of which enzyme? It is absence of alpha oxidation of branch chain fatty acid in the peroxisomes. Okay, in the peroxisomes, alpha oxidation occurs. So Refsum's disease is defect in alpha oxidation. Alpha oxidation is for branch chain fatty acid like phytanic acid, branch chain fatty acid, example phytanic acid. Okay, this is present in dairy products and green leafy vegetables. Okay, so this is defect in alpha oxidation, Refsum's disease. And alpha oxidation occurs in peroxisomes. In peroxisomes, only 2 pathways occur: alpha oxidation and oxidation of very long chain fatty acids. Okay. Next is, which of the following enzyme is used in active site with selenocysteine? So the enzymes which use selenocysteine at the active site, these are known as selenoproteins. Selenocysteine, the 21st amino acid. So glutathione peroxidase, thyrodoxine reductase, and deiodinase. All 3 are the answer, A, B, and C. So these are these are selenoproteins, we can say. They use selenocysteine at their active site. Okay, it is not ADA, adenosine deaminase, not glutathione synthetase. So what is ADA? ADA is adenosine deaminase. Adenosine deaminase. This enzyme converts adenosine to inosine. If this enzyme is increased, it is suggestive of tuberculosis. If it is decreased, it will lead to SCID, severe combined immunodeficiency, right? Next, patient with reduced levels of HDL, ABCA1 mutation, what is the diagnosis? You can see in this picture, these are enlarged orange tonsils. So what is the disease? This is Tangiers disease. And we'll do this Tangiers disease in detail. Tangiers disease, also called familial alpha lipoprotein deficiency or hypo-alpha lipoproteinemia. Which is alpha lipoprotein? HDL is alpha lipoprotein. There is decreased HDL in Tangiers disease. The mutation is in ABCA1 gene. The protein is ABCA1 transmembrane protein. ATP binding cassette, A1 transmembrane protein. This protein helps in throwing cholesterol and phospholipids out from peripheral tissues to HDL so that mature HDL is formed. So see, by giving cholesterol, when cholesterol will be given to HDL, only then the fully mature HDL will be formed. So fully mature HDL will not be formed. That's why these patients have hypo or decreased HDL. So decreased HDL, cholesterol, cholesterol esters, and phospholipids increased in peripheral or extrahepatic tissues, right? Obviously, these things are not taken up from peripheral tissues, so they will be increased in peripheral tissues. Cholesterol accumulation occurs in various body tissues. So because cholesterol is not getting, taking the efflux from peripheral tissues, so it will get deposited, like it will get deposited in the tonsils, giving enlarged yellow-orange tonsils, which is the characteristic feature. The picture was also shown, right? This picture. And there's enlargement of throat, liver, spleen, lymph nodes. So these patients have hepatosplenomegaly, peripheral neuropathy, mononeuritis multiplex is present, right? Now, the other option in here was A beta lipoproteinemia in this question. So let's differentiate this one. So in Tangiers disease, there is decreased alpha lipoprotein, that is HDL. In A beta lipoprotein, it is very clear from the name, absent beta. A beta is absent beta lipoprotein. Okay, so this is Bazin-Konsvig syndrome, also called Bazin-Konsvig syndrome, autosomal recessive and a rare disorder. A is absent beta lipoproteins, that is VLDL, IDL, LDL. VLDL is pre-beta lipoprotein. IDL is called broad beta lipoprotein. LDL is called beta lipoprotein. Defective assembly and secretion of ApoB containing lipoproteins, that is ApoB48 and ApoB100. So ApoB48 is in chylomicrons. So there will be decreased chylomicrons also. And the mutation is in MTP, microsomal triglyceride transfer protein. Okay. This protein now transfer triglyceride from intestine and liver to chylomicrons and VLDL. Okay. Yes, Vishal, I'll provide the PDF. I already told you. So this protein transferred triglyceride from intestine and liver to chylomicrons and VLDL. So chylomicrons will also not be formed, right? Defective absorption of fat-soluble vitamins will also be there then, because chylomicrons help in the absorption of fat-soluble vitamins. These patients also have neurological deficit. So in Tangiers disease also, in A beta lipoproteinemia also, this neurological deficit is present. Other clinical features are failure to thrive, diarrhea, vomiting, malabsorption of fat, acanthocytosis, that is irregularly spiculated erythrocytes, anemia, reticulocytosis, hemolysis with resultant, obviously, hyperbilirubinemia. So I hope you can differentiate between Tangiers disease, which is decreased alpha lipoprotein, and A beta lipoproteinemia, which is decreased beta lipoprotein, or absent, almost absent, negligible beta lipoproteins, right? Now, next, which of the following statement about Sirtuins is right? What are Sirtuins? Sirtuins are silent mating type information regularly protein. Silent mating type information regularly proteins. So these proteins, they increase insulin sensitivity and support glucose metabolism. Yes. They have enzymatic activity of histone deacetylase, which causes chromatin condensation here and also helps in DNA repair. Base excision repair method is used. They delay aging. Sirtuins are found to delay aging, so we can say they increase lifespan. It is stimulated by niacinamide or nicotinamide. Niacinamide or nicotinamide? No, it is inhibited. So answer is D, because question was asking except. So silent mating type information regularly proteins, they increase insulin sensitivity and they help in good glucose metabolism. And the enzymatic activity is histone deacetylase, right? There is one more enzymatic activity of ADP ribosyl transferase in sirtuins. This ribosyl transferase will convert NAD to nicotinamide. Okay, so we can say the substrate of this enzyme or protein is NAD, and the product, one of the product is nicotinamide. So product usually inhibit the enzyme, right? So by product inhibition, this enzyme will be inhibited. So nicotinamide or niacinamide will inhibit the enzyme, and NAD we can say will activate the enzyme or will increase the activity of this enzyme, right? Now, glycogen phosphorylase is activated by, calcium. Yes, glucose 6-phosphate is the inhibitor. Cyclic AMP dependent protein kinase. This is the kinase is the activator because phosphorylation will activate this enzyme, and protein phosphatase, this is the inhibitor. Okay. Give me 2 minutes. I'll just yeah. So there were a few slides missing there. So I have arranged now. Okay. So glycogen phosphorylase question we were doing. This glycogen phosphorylase has two forms, small b and small a. Small b is inactive and small a is active. This is a catabolic enzyme helping in glycogen breakdown. So this enzyme is active in its phosphorylated state. Okay, so anything which will phosphorylate the enzyme is the activator, and anything which will inhibit the enzyme is the inhibitor. Okay. So let's do the activators. Activator is liver and muscle, right? Mainly glycogen metabolism occurs in liver and muscle. So cyclic AMP causes phosphorylation. It is the activator. Glucagon hormone causes phosphorylation. Catecholamines, epinephrine, norepinephrine, they are the activators. Calcium and calmodulin are the activators. Then 5-AMP is the activator. The how 5-AMP activates is not very clear. So then let's do the inhibitors. So inhibitors are protein phosphatase, because phosphatase removes phosphate group. Insulin, it causes dephosphorylation, so that is the inhibitor. Then glucose and glucose 6-phosphate and energy ATP, they are the product inhibitors, right? Then fructose 1-phosphate is the inhibitor, which is only in liver, okay? Then next, long-term glycemic control in patients with hemolysis is assessed by fructosamine. Fructosamine is a glycated product of which of the following? So what is this fructosamine? Fructosamine is bind with proteins like albumin, right? So it is a glycated product of albumin, right? Then Zellweger disease. Zellweger disease is also defect in peroxisomes like we did Refsum's disease. So what is Zellweger disease? In Zellweger disease, peroxisomes are absent. Peroxisomes, there's a defect in the formation of peroxisomes, that all the enzymes of peroxisomes are not there. So peroxisomes are empty. They're empty so that they're called host peroxisomes, right? Now in peroxisomes, two pathways occur, alpha oxidation and oxidation of very long chain fatty acid. So both will not occur. Now alpha oxidation will not occur, so phytanic acid will accumulate. Phytanic acid will accumulate and very long chain fatty acid will accumulate, right? So Zellweger disease is a defect in the biogenesis of peroxisomes. Next, elderly patient with elevated homocysteine and normal methyl malonyl acid levels. Identify the diagnosis. Elevated homocysteine can occur in B6, B9, and B12 deficiency. Methyl malonic acid levels increase in B12 deficiency. So it is very clear that it is not B12 deficiency, right? So answer will be folate deficiency, right? Biotin is B7, okay? B7 is required for carboxylases, carboxylation reactions, right? Also in folate deficiency, FIGLU increases, right? In B6 deficiency, xanthurenic acid increases, or xanthurenic acid comes in urine, right? Yes, answer is B9. Next question. Identify the left curve given the fact that the right curve is of adult hemoglobin. Now, in this question, you need to be very, very careful that this right curve is rectangular hyperbola. Sorry, this right curve is sigmoidal. This is sigmoidal. And this curve is rectangular hyperbola. So if this left curve was sigmoidal, then we could have marked fetal hemoglobin, but now it is rectangular hyperbola. So answer is myoglobin, right? Fetal hemoglobin also shift the curve to left, but it will be sigmoidal. The curve given is hyperbola, rectangular hyperbola. So this is of myoglobin. The left side curve occurs in case of alkalosis, fetal hemoglobin, carbon monoxide poisoning, and methemoglobinemia. Right shifted curve occurs in acidosis, increased temperature, and increased 2,3 BPG. 2,3 BPG helps in the release of oxygen from hemoglobin HbA. Next exam will be May 2022 INI CET recall questions. Now, first question here is uracil was found in genetic code. Which vitamin deficiency leads to this condition? Can you tell me? Uracil is a pyrimidine nitrogenous base, this kind of ring with nothing present. So uracil is found in RNA. T-thymine with a methyl group is found in DNA, right? Genetic code, so usually should be having thymine, right? Thymine is present in DNA. Now, if uracil is there, means this methyl group is not added, and the source of this methyl group is tetrahydrofolate. So answer is folate here, right? Uracil found in genetic code, vitamin deficiency will be folate deficiency. Next, dideoxy. Here DD stands for dideoxynucleotide triphosphates. In Sanger sequencing technique uses which of the following? So dideoxy, so usually we have deoxyribonucleotides which are found in DNA. Ribonucleotides are found in RNA, right? This is dideoxy. Dideoxy always stops DNA synthesis. Stops DNA synthesis. What is a dideoxy sugar? See, this is 2' position of sugar. This is sugar, this is 2' position, this is 3' position. At 2' position, we have OH in RNA. So if we remove this O from 2' position, only H is there, then it will be DNA. If we remove O from 3' position also, this will be dideoxy. Dideoxy. So for continuation of polymerization of DNA, we need this termination, for continuation of dideoxy, we need 3' group. 3' group is required for DNA synthesis. At the end of the chain, 3' group is required. Now, there is no 3' group here, so it will stop DNA synthesis. So it uses termination of polymerase in dideoxynucleotide method, right? Then eicosanoids, they are formed from which of the following? Eicosanoids, that is leukotrienes, etc. They are formed from arachidonic acid. And arachidonic acid is omega-6 category. So PUFAs, we have omega-3 and omega-6 category. Omega-3 PUFAs are cervonic acid, alpha-linolenic acid, and timnodonic acid. Omega-6 is gamma-linolenic, arachidonic, now this another one is linoleic. The precursor of these category is omega-3 category precursor is alpha-linolenic, omega-6 category precursor is linoleic. So this means that if we have linolenic, alpha-linolenic in the diet, then it can make other two of this category. Linoleic can make another two of this category, right? So linoleic, which is 18 carbon and 2 double bond, can form arachidonic acid, and arachidonic acid is very important for the formation of eicosanoids, prostaglandins and leukotrienes, okay? Next, deamination of methylated cytosine will form which of the following? So methylated cytosine. Cytosine has amino group here. This is a single ring, that is pyrimidine ring, nitrogenous base. So amino group is there. Now, if there is methylated cytosine, okay, this is methylated cytosine. So removal of this amino group will give rise to thymine. Deamination of methylated cytosine will form thymine. So that is the answer. It is very easy, right? Also learn that deamination of cytosine will form uracil. So cytosine of simple cytosine, not methylated cytosine, deamination of cytosine will give rise to uracil, right? Then Next is you are expected to produce a protein of interest. Arrange the following steps in a sequence. So this is something related to like lysis of bacterial cell, incorporation of gene of interest into bacteria, hybridization, elution, extraction, right? So you can understand that this is recombinant DNA technology steps shown here. So we can just quickly note down the steps of recombinant DNA technology. So what we do here, we will isolate the gene of interest, isolate gene of interest will be isolated. It will be put into the vector, example plasmid. So let's say this is plasmid and this is gene of interest, put into the vector plasmid. Then this vector will be put into host, that is E. coli. Will be put into host, that is E. coli. Suppose this is E. coli bacterial cell, this vector will be put here. Now E. coli cell will do the expression of this gene and will produce many proteins. So proteins will be formed. Then we have to extract this protein. So we'll do lysis of the bacterial cell. Lysis of bacterial cell will be done. We will be getting lots of protein mixture. When we break down the cell, obviously we'll not only get the proteins from the plasmid, which are formed from the plasmid, but other proteins of the cell also. Lots of mixture of protein cell. Now we need to separate the protein mixture. To separate, we'll do electrophoresis. To separate, we'll do electrophoresis. Like SDS page is a type of electrophoresis which is most commonly used. Then to visualize, we'll do blotting. For proteins, we do which blotting? Western blotting is done for proteins. And then elution means extraction of these proteins can be done. So these will be the detailed step sequence in recombinant DNA technology, right? Next, DNA protein interaction can be studied by using which of the following? DNA protein interaction is done with the help of DNA footprinting, right? So DNA footprinting analyze protein DNA complexes and identify the position of the binding site as well, right? That's why footprinting, as if footprints are marked, right? On the other hand, fingerprints are very unique. They are unique portions of DNA for the very particular individual. You know, fingerprints are very unique, right? Northern blotting is for RNA. That cannot be the answer here. ELISA is for antigen antibody reactions. That cannot be the answer here for DNA protein interactions, right? Okay, next. Which of the following is false regarding Human Genome Project? So this Human Genome Project was started in 1990 and was completed in 2022. The Y chromosome was fully discovered in 2022, fully decoded, right? So it was done for almost 2 decades, right? So there was collaboration of many countries, yes. Around 20,000 genes identified, yes. It uses modern sequencing techniques like next generation sequencing, which is quite an automatic system of DNA sequencing, very fast and automated, right? It took you 2 years to discover? No, almost 2 decades. So, which is false is option D, right? Next is November 21 questions. Most preferred chromatography for HbA1c is ion exchange chromatography, right? To be very precise, it is cation exchange one, okay? So you can just cram this one. Which of the following is not glucogenic? Substrates of gluconeogenesis are very, very important, and this full topic of gluconeogenesis is very, very important. And we all know that acetyl-CoA is never glucogenic. It is again and again written, acetyl-CoA can never be the substrate of glucose. So answer is not glucogenic is acetyl-CoA. Also, acetyl-CoA activates this pathway of gluconeogenesis. It is not the substrate. It cannot form glucose, but it is the activator of this pathway, gluconeogenesis. It activates which enzyme? The first enzyme of gluconeogenesis, that is pyruvate carboxylase. Okay. Oxaloacetate, pyruvate, lactate, all are substrates of gluconeogenesis. So we can just do substrates of gluconeogenesis. One is pyruvate and lactate. Other is glycerol and propionic acid. Another is amino acids. Amino acids, that is glucogenic category amino acids, and both category, both ketogenic and glucogenic amino acids, right? Okay. Next is thiamine is a coenzyme for. Tell me the answer. Thiamine is a coenzyme for. Okay, Godavari, you are asking that in Human Genome Project, what was the sequencing used? It is called next generation sequencing. Next generation sequencing can use any of the method, right? It is just the automatic version of our chemical method, right? Or dideoxytermination method can also be used. So that is not important, but next generation sequencing method is used, right? Now, next is thiamine is a coenzyme for. Thiamine, that is B1, is a coenzyme for pyruvate dehydrogenase complex, that is link reaction enzyme. It requires B1, B2, B3, B5, and lipoic acid. Total 5 coenzyme. Lipoic acid, total 5 coenzymes are required. Okay. Then pyruvate carboxylase, it will use B7. Then PEP carboxykinase, PEP-CK, or any kinase will use magnesium, right? Mostly kinases uses magnesium. Pyruvate kinases uses potassium more than magnesium, right? So these are the few questions repeatedly asked. CRISPR-Cas9 repair method is associated with. It is associated with non-homologous end joining repair. Right? This is to be crammed only. Just cram this one. Okay, there are a few questions which are at the level of research, right? So those questions are also asked in INI CET exam, and mostly these questions are not rank deciders because all the students will not be knowing them, right? And when we are doing this question as revision, then also we have just read some research papers and then we have taken out the answer. But the things are still in research, so we do not know the explanation, the mechanism behind it, right? So it is better to cram such questions. It will help you simplify the subject and you have to revise all the subjects, okay? Then, which require maximum energy for assimilation. Maximum energy for assimilation or absorption. So this is proteins. Proteins are high on thermic effect. This is also called thermic effect or specific dynamic action. This is maximum for proteins, then for fats, and then for carbohydrates, right? Okay, then true about mitochondrial DNA. True about mitochondrial DNA. It has high chances of mutation as compared to nuclear genome. Yes, because there are no introns present here. Presence of introns prevent mutation. So when introns are not present in prokaryotes also, they're not present in mitochondrial DNA also, because this is same like prokaryotic DNA, right? The eukaryotic DNA, which we call is that is present in nucleus. Okay, it is single-stranded circular DNA. It is circular, double-stranded circular DNA. It encodes all ETC proteins. No, it encodes 19% of ETC proteins. That is total 13 out of 67 proteins, right? So this is wrong. This is wrong. It has around 1600 base pairs. No, 16,000 base pairs. So answer is the first one. It has high chances of mutation as compared to nuclear genome. Vobel hypothesis is due to which property of codon? Vobel hypothesis is due to degeneracy of codons. The third base of codon, so this is suppose mRNA, 1, 2, and 3. This is 5' end of mRNA, this is 3' of end of mRNA. So this is the third base of codon. Codon is present on mRNA. And this is tRNA. So tRNA will be 5' here and 3' here. This will be first, second, and third. First, second, and third, right? Now, this is Vobel. The third base of codon binds with first base of anticodon with non-specific base pairing. That is, it does not follow Watson-Crick base pairing rule, right? Then July 2021 questions, microRNA, very, very important. MicroRNA or RNA interference technique or silencing technique. You have to revise this topic. This is very, very important, right? Whatever I can take in this short session, whichever is possible, that I am taking, right? Otherwise, you take it as a stimulatory session for you to read, right? And you can ask your doubts in the Telegram group also, okay? So microRNA binds to mRNA on which site to inhibit translation? It binds to 3' end of the mRNA, right? 3' end of the mRNA. Now, this is also called gene knockdown. So there's one more question on this topic. Let's continue further. What is the effect of RNA interference on gene expression? Yes, this is another question. The effect of RNA interference or silencing technique is gene knockdown, right? Gene knock in means gene is inserted, a new gene is inserted. Gene knockout means gene is deleted, right? So these are not very successful techniques, but gene knockdown is very, very successful and quite frequently used in research in molecular biology, right? So this is very, very important topic, right? Here what happens is that there's a cell, and if this cell, so this is nucleus, So if this is DNA, DNA will make mRNA. This mRNA will come out here and will make protein in the cytoplasm. Now, microRNA, I'm going to make in red miRNA, microRNA. It is a small size RNA. It will go and bind to the 3' end of this target mRNA, and it will either suppress this mRNA or will degrade this mRNA so that protein synthesis cannot occur, right? Protein synthesis cannot occur. So the gene is still present, but it is suppressed. Okay, so gene knockdown. We are not touching the gene. We are not even touching the gene, but we can create a synthetic microRNA to inhibit the target mRNA so that we can inhibit the translation. Okay. Which of the following enzyme and its diagnostic marker is incorrectly paired? Very, very easy question here. Amylase lipase for acute pancreatitis. This is correct. Ceruloplasmin and hepatolenticular degeneration. Wilson's disease. That is correct. In Wilson's disease, there is decreased ceruloplasmin. Do revise Wilson's disease and Menkes disease. This is very, very important. In copper, the two diseases, Wilson and Menkes disease. Okay. Then in von Gog's disease, which is glycogen storage disease type 1, which enzyme is deficient? Glucose 6 phosphatase. Von Gog's disease is frequently asked, very, very important. Okay. But the enzyme written here, beta glucocerebrosidase, that is of Gaucher's disease. So this is incorrect. Then LDH1 and LDH2 ratio in myocardial infarction, there is flipped ratio of LDH in MI, right? Then insulin inhibits which of the following? Insulin inhibit catabolic enzyme. Lipoprotein lipase is anabolic enzyme. Hormone sensitive lipase is catabolic. So answer here is hormone sensitive lipase, right? And insulin activates lipoprotein lipase. So these two enzymes are very, very important. The first video, first and second video of my sequence in videos, that is very, very important to understand. So many questions on enzymes and phosphorylation, dephosphorylation, hormones. So the formulas which I've given you, you can revise that. Okay. Then cytochrome oxidase, that is complex 4 of ETC, is inhibited by all of the following except hydrogen sulfide, yes, carbon monoxide, and cyanide. They all are inhibitors of complex 4 of ETC, but not methane, right? Then what does the graph represents? This graph is very clear, very easy question, Michaelis-Menten graph. It is Michaelis-Menten graph. So it will show Michaelis-Menten kinetics, that this is rectangular hyperbola graph. So this is Vmax point, this is half of Vmax. If this point, half of Vmax, is extrapolated towards x-axis, then this value is Km. Km is Michaelis-Menten constant. Km is that substrate concentration at which velocity of reaction is half of Vmax, right? In fasting, RBCs utilize. Tell me the answer. In fasting, RBCs utilize. Yes, dear, my lectures will be same in all the platforms. In fasting, RBCs utilize glucose. Whether it is fed state or fasting state or starvation state, RBCs are on glucose, right? And in RBCs, 90% of glucose will be giving 2 ATP per glucose. 10% glucose of RBC will go into RL shunt, where no ATP will be obtained, but 2,3 BPG will be obtained, right? So this is also frequently asked and very important. Then increased methyl tetrahydrofolate accumulation occurs in deficiency of which enzyme? Methyl tetrahydrofolate gets converted to tetrahydrofolate with the help of vitamin B12. So in B12 deficiency, methyl tetrahydrofolate will be accumulated. Answer is B12 deficiency. Next, which of the following method cannot be used for protein precipitation? Protein precipitation occurs at isoelectric pH, PI. Isoelectric pH. And many agents like heat, acetone alcohol, heavy metal ions, trichloroacetic acid, they are used. Also sulfosalicylic acid, they can be used for protein precipitation, right? So first option, alcohol and acetone, that is correct. Trichloroacetic acid and metal ion, they can be used. But moving pH away from isoelectric pH will give some charge to the protein. And at isoelectric pH, there is no charge. That's why precipitation occurs. Okay, so answer is this. See, G1 phase, this is not possible to revise the formulas in this class. This is a short revision of PYQs here, right? So let's just cover these here. You can watch my videos anyways. So this is it. You can ask all the doubts on DBMCI Premier Group on Facebook and Telegram. So the link will be shared in the chat section. So this is it with this section. Thanks for watching. Best of luck for your upcoming exam. Do work hard, wake up early, eat less, because when you eat less, eat more of fruits in this summer season, then it will help you a lot in concentrating more, sleeping less, and focus more on your studies, okay? So this is it. Success is not final, failure is not fatal. It is the courage to continue that counts. So I hope you all will keep continuing in life and do never, never give up. Bye-bye. Thank you.